Right. Gene, how's it going, man?
It's going pretty good. Just got back from the old UNPA member retreat in Utah. It was beautiful there. You saw some of the pictures.
I saw those pictures. Yeah, gorgeous up there.
To a lot of interesting presentations, and yeah, it was pretty cool.
Cool. Good time of the year to go too.
It was.
Yeah. Well, welcome everyone. We didn't do our podcast on Friday because Gene was out for the UNPA conference, so we're catching it up today. So the topic today is as a comprehensive review of botanical identity testing, and I just published and wrote an article about this. Was that last week? Yeah, I guess it was.
Yeah, it was.
Last week. Anyway, so that's the topic
today.
Good
article too, guys. You should read it. It's very interesting. Tell them where they can read it.
You can find it on my website at natprologics.com. It's in the blog. Also, if you check out my LinkedIn profile, we're connected on LinkedIn. You can go see a post there, and various places. But if you can't find it or whatever, just let us know. I'll put this in our posting when we finish the episode here and broadcast. I'll put the link again for everybody to go read the article.
So here's something I wanted to let everybody know. If you've watched our podcast before, you know that Bill and I have a topic. We kind of go back and forth on the topic and more or less play an equal role in doing so. But in this particular case, the botanical identity testing, I insist on deferring to Bill's expertise. Besides the fact that the man has a PhD in natural chemistry, I mean, his dissertation was on analytical testing methods. This is really, really an area of expertise for him. I have some knowledge of this, but not to the depth and breadth that he does. That being said, I have some questions for Bill that I want to bring up. So I'm going to lead this discussion by asking some of those questions. But just before doing so, I'd like to give you guys a sense of why are we doing this, comprehensive review of testing for botanical identity? Well, let's just consider the botanical market size. Right now, the global size for botanical supplements by the end of this year should be around 31 to 32 billion US dollars. That's a lot of dinero. And then if you just look at the projection, just to 2029, it's projected to reach 50 billion. This is a very, very meaningful category. Consumer awareness about the health benefits of plants is huge. You see this whole shift that's taken place, preventive health care, popularity of natural remedies, looking at things like, anxiety, menopause, insomnia, just the many different things that somebody might use a botanical and herbal for. And so, this is just such a huge thing. And now if you start looking at food and beverages and personal care, that's expanding as well. If you include some of these other things, now you're looking at 175 billion just last year, and further growth to 281 billion by 2023. So again, very, very significant thing to look at here. So that's why we're doing this. Now, that being said, let me start with my first question. Okay, Bill, so let's talk about the first sort of category of questions. First category of questions is going to be just a little bit about the background on botanical identity testing.
Yeah.
So I'd like to know, can you give an explanation of really what the botanical identity testing entails, some of the historical methods used, and maybe a little bit about practices such as macroscopic and organoleptic analysis. Just so what it entails, some of the historical methods and modern and then macro and organoleptic. So take it away, Bill.
All right, man. Pleasure to do that. I think I want to put a context or framework around this whole discussion because there is no one-size-fits-all solution. That's really important take-home message today. And it really depends on the botanical, the class of compounds that you're trying to test and the botanical you're trying to identify. There's actually multiple variables in terms of that. And we'll get into a lot of those details today. But for sure, botanical identity testing's been around for a really long time. And what does that mean exactly? That means it basically identifying the plant for its, what genus it is, what species, sort of thing. This goes right at the beginning, right to the farm before you even do any kind of analytical type testing methodology. But nobody had before modern chromatography, which is the main principle. We're going to talk about various chromatographic techniques today, but before the advent of modern technology and chromatography like that, how did people identify botanicals? And usually traditional healers or shamans or herbalists, they passed on their ethnobotanical knowledge from generation to generation. It was an oral tradition, and in terms of that. And there are a lot of examples, Gene, historically. I only listed a few in my article, but just a couple examples like in ancient Egypt around 1550 BC. You have the Eber's papyrus, papyrus. I think that's how you say that. You have the Ayurvedic texts from India around 1500 BC. So we're talking thousands of years, really.
Yeah.
And even in my own ancestry as a Native American Cherokee, this goes back to my great grandmother and beyond. How did the native indigenous cultures in various countries would identify plants, and they would do that by observing animals and their behaviors and stuff. I always wondered, Gene, who was the first person that taste tested a botanical? What if it was poisonous?
Mm-hmm. Well, that's why they look at, that's like you say, they look at animals, and there was this whole doctrine of signatures concept where if it looked this way, it must be good for that, and so people had a whole concept of it. But I think just trial and error, and some bad cases of heartburn later, people started figuring some things out.
Well, and if you think about it, that it was all observational, and in fact, we still use these types of techniques in our modern healthcare, and so these rich traditions were actually the foundation for pharmacognosy, and also led to the development of the first drugs from plant sources,
Mm-hmm.
back in the 1800s. So it's still a valuable resource today. Although I would say this tradition, the historical knowledge that people have of plants, there's a few folks like that have that sort of thing in our industry, as you know, but not a huge number of people are aware of that. Everybody goes right to the analytical methods we have available in modern technology. But anyway, and of course, don't forget Chinese, traditional Chinese medicine. It's been around for thousands of years as well. So there's been a lot of publications of that over the years. And that really was the basis of our modern drug programs and stuff like that. I don't know if you know, but post World War II or during World War II is when penicillin came about, and it was like a miracle drug, So and it came from mold or whatever. So not a botanical, but again, natural sources or natural products. There's tremendous number. It's a treasure trove of chemistry in the natural world, and we don't even have know a fraction of what those bioactives are today still.
Well, and then just we're talking macroscopic and organoleptic. I can walk out into my garden, and just because I see the plant, I see the leaves, I see the stem, I was like, oh, well, that's echinacea. Of course, my wife planted it, so that helps. But you can see it. I can see some things. Oh, look, it's got the square stem. That's in the mint family, blah, blah, blah. And you could, in some of these things, you can taste safely and distinctly be able to tell. But you have to have training to do that.
You have to have training. It's one of the qualitative methods that are still valuable today, So, if you're a product brand producing a product, obviously you're not out at the farm doing that. You're getting your ingredients from some other ingredient supplier, who in turn is probably getting it from someone else who's a distributor from wherever it's grown. And so, this kind of farm to table concept in terms of that. And, this is just kind of an aside, Gene, but, the whole sustainability issue and stuff, supply chain is a huge logistical problem for our industry. It has lots of challenges, and think about that for growing plants. And, we've had issues over the years where we've had issues with supply chain based on, supply and demand economics.
Yeah, well, so let's have that be the next category of
Yes.
questions.
Okay, next category of questions.
Baboom. Okay.
Baboom. Yeah.
So challenges. So there are challenges in botanical identity testing.
Oh,
So
yes.
can you address some of the complexity, the variability of botanical ingredients, things like species, plant parts, etc., and common issues, things like adulteration, contamination, challenges that these present to reliable and accurate testing?
Sure. So again, it's really important to know what you're working with right from the beginning. What part of the plant, genus, species, all this kind of stuff. Anything from literature, if is available in terms of research that's been done on plant, that sort of thing, in terms of the class of compounds that are present, whatever. But there's really three kind of major challenge areas when it comes to analytical testing of botanicals. The first would be just simply due to the complexity and variety of botanical ingredients that are available. So, for example, you may have turmeric's a good example of this. You may have turmeric, root, and the curcuminoids is the class of compounds. And there's about 300 plus compounds that are in that class that come from the root. And so, but people aren't harvesting the leaf or the flower or seed for that. They're harvesting the root. And that's where turmeric, which is a traditional spice, it's been around a long time. It does have some benefits there. But you could have a whole spectrum of botanical materials. I mean, you could have just a ground up root powder. You could have a native extract without excipients where they've done extraction. And it could be extraction with water, could be supercritical CO2, whatever the extraction method is to pull all those goodies out of the plant part. And then you could all the way going up to as you purify and do different chromatographic methods to purify it. I mean, if you look at it's very complex in terms of you've got could have several hundred components in just a plant.
Oh, sure.
And it'd be different from the leaf to the flower or the seed or the root. I mean, they all have a little bit different chemistry. But there'll be different classes of compounds sometimes in those things. So you really have to know what botanical material you're working for and understand that you can have. So if you just see something that says a certain botanical on the market, you need to look at what part of the plant it is. And there's traditional use for certain parts of the plant. If we look at ashwagandha, for example, the root is what's used for traditionally Ayurvedic medicine, not the leaf, not the seeds or anything else. That this doesn't
traditional use. That's absolutely true. And now you find some extracts out there that some companies have put out, and they've done some science on it. And they've done some studies, and they've shown, hey, if you combine it with the leaves, you get these effects, whatever, and that's fine as long as you've got the safety data, as long as you've got the science that supports it. Certainly not the traditional use. But and then of course you have stuff like the region of the region it's grown in. Man, that'll make an impact, right?
Yep, it will. You'll have seasonal variability. I'll talk about that with a personal experience on a case study at the end here, but that illustrates that sort of a scenario. But the second challenge, the first one was kind of the materials you're working with. Second challenge has to do with adulteration or contamination. Okay?
Yeah, that's a big deal these days. American Botanical Council has a whole program on adulteration.
Yeah. Right, and that's either can be intentional or non-intentional. Actually, it happens. It happen either way. But some people that are producing the ingredient, the extract, whatever, can inadvertently contaminate or adulterate it, and it can be done by other plant materials being added, or fillers, or synthetic substances. We've seen all that in the industry over the years, right?
Oh yes.
How many times have we seen the men's sex products that actually had Viagra or Cialis or a component in there?
And those were tested and found and rightfully removed from the market because that's a dangerous practice.
That's a dangerous practice.
And like you said, I think there's sometimes people that there is adulteration and they don't necessarily realize it, but there's times when it's fully realized. Again, not going into case studies, but I can think of times when, you had something like a Hoodia back when,
Oh
where
yeah.
there was more Hoodia on the market than there was actually grown in the world. It's like, oh, really?
Yeah.
Yeah, that's probably not all Hoodia then. It's probably
It's
other stuff.
probably not all Hoodia. And most recent example, right, happened during COVID, right? We had in 2021. Let's see, was it 2020 was elderberry was the number one selling botanical ingredient because it was all
Yep.
about immune health, right?
Right.
Well, of course, that opened the door for some bad actors to adulterate of course, the price for elderberry went up. I remember walking into a vitamin shop and there was a whole shelf space dedicated to elderberry products. Okay. I mean, it was like amazing. And, but this is just one example that because of supply and demand economics, the opportunists, want to join in the market, and it became very difficult to get supply of it. And of course, people just adulterated and called it elderberry or whatever. I think the statistic was 30% of the elderberry products or ingredients on the market were adulterated. So at the
That's
time,
crazy.
that's crazy.
Absolutely nuts. So,
So,
yeah.
so our kind of our other third key challenge, okay, has to do with the botanical sources. Okay, so you can have genetic diversity, all right, within different species, subspecies or varietals. Think about grapes for winemaking, right? You have different varietals of the grapes there, and the chemistry is quite different. It's very complex chemistry. I know, like, for example, a traditional Cabernet have like 450 components in it, right? And that's not just from the grape itself, but that's from the fermentation process and from being aged in oak barrels, and it leaches out what's in there, those chemical compounds. That's what gives it the complexity of flavors and tastes. I actually admire the guys that are some sommeliers.
Oh,
that
yeah.
that can detect. They're like a human gas chromatograph, or you know what I mean, in terms of tasting volatile.
I taste high levels of apricot with some pepper undertones, and I'm like,
yeah. I'm not that sophisticated. My palate's not sophisticated. I remember seeing a research article, though, where those people that can do that have a larger olfactory setup in their nasal cavity, and they're able to. It's like a gas chromatograph in a way. Those volatile compounds. A lot of them are volatile, so that turn into vapor. They can sense that. So pretty cool. Pretty cool. But this is kind of what you were talking about before: geographical origin, environmental factors. Affect the growing, the soil type, the temperature, humidity, rainfall. So anybody who's an ingredient supplier in our industry, Gene, should really be concerned about this factor and where they're getting their source. And there are some examples of ingredient suppliers who are vertically integrated, meaning they control the supply chain right to the farm.
Yeah.
Okay. And actually KSM 66 Ashkelon is a good example of that. They have their own farms, and they control the growing and the quality of the product. So they are doing it in a very sustainable way. And you do have variations. They have variations from crop to crop, and they're able to standardize and account for that sort of thing. But the real life example of this, I'll talk about later, is just remind me to talk about St. John's Wort when we talk about the case study below.
Okay, I'll do that. So, shall we move on to the next category?
Yes, let's do that.
Okay. So, we have both qualitative and quantitative testing methods. I'd like to start with qualitative and ask you to go and give an overview of that. What are some of the different methods, the pros and cons, and some of the importance of expertise and experience?
Sure.
So
we'll start with qualitative, but I think we need to define what is qualitative and quantitative.
Very important.
Some people don't know that. So qualitative is a method that's very subjective. Okay, it's open to interpretation. All right. So, for example, looking at the leaf or whatever, this is where the expertise comes in. When they're looking at the plant, they can identify the plant. Oh yeah, that's echinacea. I can tell by the leaf and the flowers, right? Where versus quantitative is objective, it's statistical, numerical. Okay, so there's a statistical analysis. There's a measurement of the components actually in there physically of whatever compound A, B, C, D is in the plant, right? And there's ways to do that. Now, qualitative method can't do that. So quantitative, numerical, qualitative is more visual observatory, right? So it's open to interpretation. So obviously, we mentioned this earlier. You mentioned it, macroscopic analysis. That's a specialized field. People who are botanists or plant experts, they can identify plants by visually looking at them, and they know the shape and the color. There are modern, herbalists, from that have actual training in that. And as part of that is organoleptic analysis, and that has to do with taste, touch, and smell to identify the plant. And then, of course, there's pros, and we'll talk about this each one. We'll just briefly do this, but then I invite people to go back and read the article in detail. They can read through it, all the examples. But the pros for that, these types of analysis are simple, it's cost effective, but it is highly subjective. It's limited to non-powder forms. Obviously, you can't do it on a powder and identify what the plant is, right? Because it's already been processed. And it requires, one of the limitations requires expertise. You'd have to be a botanist, someone that really knows plants to do this sort of thing. Same thing for microscopic analysis. That's qualitative as well, but that's using a microscope, a little bit modern technology, but you're looking at the microscopic level, you're looking at structural information, and this is useful for powders. You can apply this to powders in addition to the plant. But again, it requires specialized equipment and training. Again, you have to be trained to do this sort of thing. So it's not like, I think it's important to call out, Gene, that you can't just send any kind of sample, anybody, to any lab and expect them to be able to test it and do the identification for you. No, and we'll talk about this towards the end here, but you really have to know your stuff here a little bit, because you don't know what you don't know. You got to ask the right questions when you do that. But again, it's limited because it's time consuming, whatever. And then our last qualitative method is TLC, or thin layer chromatography. So this is a very simple qualitative method that if I just describe what chromatography is in general, because this applies to some of the quantitative methods as well. You have hundreds of compounds in a plant, right? And you may have extracted them with water or methanol, supercritical CO2, whatever the method is, you're going to do that and then do further separation and so on, depending on what type of extract you're producing. And it's based on polarity. So you know what polarity is. It's like positive and negative charges. So different compounds are more polar, which, think about salad dressing, right? So you have oil and water, right? So those compounds that are very polar are going to have an affinity to be in the water phase, and those that are very fat sensitive, lipophilic, will be in the fat phase, in the oil phase. It's the same thing with plants. When you're doing these extractions of natural products, you're separating based on these polarity differences to get to purified to different sets of classes of compounds. And then there is a high performance thin layer chromatography, HPLC. It's a little more sophisticated, but the main distinction is that you have more absorbent particles. You have basically a glass plate or a plastic plate that's coated with an absorbent material, okay, like silica gel. And so different compounds with a solvent it's going to sit in a solvent that travels up. You've seen this capillary action of things going up paper or something like that. It's the same principles, but any of those chemical compounds in the plant are going to separate by polarity, so that silica phase or whatever that material is on the glass versus the solvent have different polarities. And so that's going to separate these things on the plate. That's probably more detail than people want to know, Gene. But now, some of the pros of this, you can do a visual comparison. And there's reagents to use, like UV light and staining reagents, where you can, knowing the class of compounds in the plant you're trying to identify, you can see that, because some you can't see visibly with the naked eye. So you have to have some help there. But one of the cons for this is that it has limited sensitivity, and it's semi-quantitative at best. You can't quantify the amounts of different components in there. And sometimes, if you just on the principle I just talked about, you can have different classes of compounds that have the same polarity that come off, and you see a spot on the plate, but that could be more than one component there. right? So you can't quantitate that and necessarily identify what's going on there. So, and again, it takes experience to do the interpretation. It's very difficult. I can't tell you how many times I've done TLC in the lab trying to separate things or whatever. It's very
So would
complicated.
you say that the main benefit of qualitative testing is just, let's call it plant identification, more or less? You know, you're saying, yeah, that's what this is, as opposed to any kind of, and it contains this percentage of whatever. You know?
Yeah. And you're actually, I didn't say this, but you're doing this against a reference standard. So you know something that's in that plant, a certain compound,
right?
And it's on the plate next to it, and you can see it going up the plate at the same point, and see a spot on the plate, and you say, okay, it has that in there, so this looks like it's that. But again, you can't rely on this as a totally quantitative method. It would be dangerous to do that, because you're going to have misidentification. I mean, just what we talked about before, you could have the actual, let's say a fruit powder. We talked about elderberry. You could have the fruit powder, and then you can have an extract of elderberry that's going to have a different chemical profile, and you can't compare those on TLC, for example, right? You're going to get different results because the concentration is different for the components in there.
So I just wanted, for the benefit of the listeners, I wanted to let them know, you mentioned a reference standard. I just want to let them know what that is. So, like, if you have a reference standard, for example, of a ginkgo extract, and you know, it's going to have certain compounds, terpenes, lactones, various actives, markers, and you can use that standard, just as Bill said, and you can compare it to the material that you're testing. Right. Where do you get those standards? You buy them. There's places like Chromadex and places like that where you actually buy those standards. Some of them aren't too expensive. Some of them are very expensive. You can pay well over $1,000 for some standards. Others a lot less. But you have to have that, because you have to compare it to. It's not like there's a machine, you just toss it in and it goes bing, 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 bing. Here it is. Here's what you got. No, you have to compare it.
No, for most of these methods, anything chromatography, you're comparing it against the standard. So, you know, for example, St. John's wort, total hyperisons, hypericin compounds, you can test it against that to see that it actually has hyperisins. But again, it may take more than one method to identify the botanical, because, like I said, adulteration occurs. I mean, people are pretty sophisticated with this gene. They can add another material that will give that result, but it may not be fully the material there. So it really takes experience to do this sort of thing. And when I started this gene, when we were doing this way back, and I'll share the story with Centrum Herbals that was going back to 97. There weren't any testing labs. Alchemist labs, all these other labs that exist today didn't exist then. We had to isolate our own standards. You couldn't even buy the standards at a chemical compound company. Okay, so we had to isolate. And you don't want to know why some of those standards are so expensive, because they're really difficult to isolate, okay, and purify. It's super hard to do. Many natural products are very difficult to do that. So it's not as easy as just pulling something off the shelf and just send it to lab and get it tested. It's so much more complicated than that.
Exactly. So don't bellyache too much when you're spending a few bucks on your testing. It's a lot involved. Let's go to the next one.
Yeah, let's go to quantitative methods.
Quantitative. Now. Okay. So all right. I want to hear about some of the different testing methods, the quantitative testing methods, and the advantages of one over the other, and other considerations like cost and so forth.
Yep, sure. So when you guys get a chance to go back and read my article, there's a nice table that summarizes side by side all the methods we're talking about today. It's a good reference table that you can go back and just double check. So HPLC, high performance liquid chromatography. Well, this is probably the most popular, most sophisticated analytical technique and probably the ones used the most, okay, in terms of quantifying individual components. So, I mean, this is in the natural products chemist repertoire. This is kind of our go-to thing, but before you even get to HPLC, you can't just take a raw extract and run it through HPLC. because you
Mhm.
have too much interference. Like I said, there's complexity in a lot of these botanicals. We have multiple compounds in there. So, in my research at university, Gene, we didn't get to the HPLC stage till much further down the chromatographic trail, so to speak, because we used big column chromatography. We use extractions based on polarity, like making our oil and vinegar separations, so to speak, and all that to get into cruder is more simpler fractions, and then that would dictate what type of HPLC method we would do to purify. We were looking for purified compounds and isolating pure compounds, and we used NMR. We'll talk about that in a minute. But anyway, so it's highly sensitive. It gives you quantitative data. It does require expensive equipment, and again, requires expertise. You got to know how to run one of these things. You can't just do it. Uh, things are A lot of things are automated today, Gene. People set it up overnight and then put their sample preps, and sample prep's important. I should call that out. Sample prep in all these methods is extremely important when you're trying to identify something. If you don't prep the sample appropriately, and that means dissolving in the appropriate solvent to solubilize the components in there, well, then you're not going to see them in a TLC or an HPLC or whatever. They're not going to be there. So it has to be soluble in that matrix. And this is very sophisticated. Actually, part of my PhD dissertation was I had developed a solvent triangle scenario for natural products, and this was marine sponges, of using a mix of 3 or 4 tertiary or quaternary mobile phases. Mobile phases of the solvent, organic solvent you're using, with various polarities to get really incredible separation in HPLC. So that was a lot of fun to do. And so again, sample prep is really key. What you use to run an HPLC in terms of the mobile phase, which is the solvent you're using, is going to give you the right separation and be able to identify there. So again, I said here the limitation: sample preparation can be complex. So there's no question about that. Now, gas chromatography, very similar. The only difference really is that you're analyzing volatile compounds. So this is quite limiting because most of the compounds you find in plants are not volatiles. All right. And if you go back to what we were talking about, organoleptic, where you're smelling something, if you smell like mint leaf, you're smelling that particular compound. But not all the compounds in the plant will vaporize, and that's what you're going to smell. Then think about winemaking, all the volatiles there. Okay, so gas chromatography is useful. Let's say you're working with a plant that has a lot of terpenes, that sort of thing. But it's only good for volatile compounds, right? So that's the limitation there. And it again requires specialized equipment and expertise. Cruising through this, I don't want to weigh in on too much detail on all these. It's very complicated. And I've used all of these techniques in one scenario or another, depending on what I was doing in my research. Mass spectrometry. Okay, again, another quantitative analytical technique. And this helps you determine the mass of the compounds. Now, a lot of people don't really need to use mass spectrometry to do botanical identity testing. I mean, we did it because we were isolating purified compounds, and they were unknown. We wanted to determine their molecular structure. So part of that is knowing the molecular mass of the compound that you're looking at. So mass spec is for that. It's highly sensitive, highly specific. Super expensive, by the way. Not everybody has a mass spectrometer in their lab, that's for sure. And it's a high level of expertise. The data interpretation is super complex. In fact, it wasn't my area of expertise, even though we used it. It was still required somebody else who actually knew how to interpret the spectrographs of that, because what you're doing is you're ionizing the molecules in the sample, and then they basically break apart into fragments. So, and the fragment will have a certain molecular weight. And by doing that, there's certain key fragments that will always fragment when you ionize the compound. And that's a good way to identify that it has that class of compounds in there. But again, you don't go straight to mass spec, Gene. You're going to do some of these simpler things first, TLC maybe, or HPLC before you want to, if you have to get more information.
But by the way, for our listeners, when Bill says some of these equipment is expensive, I used to work for a contract manufacturer years ago, and we had a very nice testing laboratory. And I remember the first time I went in there, the head of the chemist who headed our quality control department there was taking me around, showing me equipment. I'm like, oh, that looks expensive. How much was that? Well, that was about $100,000, and this one over here was about
Yeah.
$75,000, and this one is like, holy mackerel! When I looked at everything here, I thought there are millions of dollars of testing equipment in this lab. It's
Yeah.
amazing.
Yeah. Well, let's talk about our next one, NMR, or nuclear magnetic
Oh, for
resonance.
real expensive.
When we talk really expensive, oh my gosh, we're talking a couple $1,000,000 for an NMR machine. Okay, it's a big superconducting magnet. You have to keep liquid nitrogen, keep it cool, and so this is my whole thesis is on NMR because we were in the university we were the NMR jocks, okay, so to speak. So we basically were using NMR to determine the structures and compositions of compounds, and it's a very highly specific method. I'm thinking, Gene, most people don't need to do NMR for botanical identity testing. I mean, we really only use that after we've already done crude separation with other chromatographic techniques, HPLC, column chromatography, whatever, before we got down to a pure compound. But it could be useful if you're looking for new compounds or you want to quantify them in that. But it's not the first thing you go to and do in terms of that. It gives you detailed molecular structure information. In my 5 years doing my PhD and my research, I identified 8 new compounds that were not, and published about those and determined their structures. I remember one was really a big structure. It was really complicated to solve. It took 4 months to figure out the structure. So it can
Wow.
be very, very time consuming and tons of, like a stack of spectral data, of various different experiments to try and figure out what the structure is of that compound. So again, this is highly specialized, not really suitable for all compounds. And you certainly can't do anything with it unless you've done purification of your extract before you start trying to test it.
But these quantitative testing methods, this is what you have to do if you're trying. I mean, so for qualitative, let's say your company got in a sample of ginkgo, for example, and you did, qualitative, you could go, okay, yeah, this is ginkgo. But if like, okay, this stuff's supposed to be standardized for 24% terpene lactones, you can't verify that with qualitative. That's when you have to go to the quantitative. You've
Yeah.
got to use those methods in order to find out. And it is more costly. And it
Yeah.
does require that specialized knowledge, how to run these things, how to do that. But it's a necessary aspect to this type of analytical testing. And so whether you have an in-house lab or you have to send it out to a third-party lab, it has to be done every time.
And it's important actually to call out these methods need to be validated. There's an actual
Yes.
set procedure to validate a method using HPLC, for example, in the lab. And if you sent that sample to another lab or inside your own lab on a different piece of equipment, you would technically need to validate that as well to make sure it's reproducible. You can't just take it and test it in one lab. It has a validated method, and then now let's send it to another lab. You have to do an intra-laboratory, intra laboratory validations in those scenarios. And there's a proper procedure to do that, procedure with HPLC, for example. So anyway, that's really complicated. Now let's, Gene, I'm excited to talk about the next section here, and that's all
let's move there.
Let's talk about.
So let's talk the hybrid and emerging testing methods.
Some cool stuff out there. Yeah,
there is some cool stuff. Some of them are in our industry has also been controversial. Things like DNA barcoding and some of these others. Well, let's get into that a little bit. Talk about some of the different hybrid and emerging technologies, and the opportunities they may offer.
Yep. So let's talk about a quantitative hybrid method, and that's using the methods we talked about before and combining them in tandem. Okay, so HPLC MS, so high performance liquid chromatography and mass spectrometry. So basically, you have an HPLC hooked up to a mass spec. You put your sample in, and it goes through, and you get your separation. You see that, and then when it goes, because it's coming out at different time points and being eluted at different time points, then it hits the mass spectrometer at different points. So this helps you to kind of do some more sophisticated, very specific data analysis here. And again, a GC mass spec, same principle, but again for volatile compounds, and then using that in tandem with mass spec. So these aren't new, Gene. These have been around. I mean, I was using those 25 years ago. So, these types of techniques have been around. They've got better technology and better ways to computers to analyze the data and stuff.
I was doing some searching a while back through PubMed, looking for some specific types of testing methods that involved NMR, and I found it almost always in combination with something else. Talk about a hybrid method that was almost constant.
And so again, you need to have the expertise to do this stuff, and most labs don't have people that have this sort of expertise. And it's not in natural products chemistry. Again, these more advanced techniques are generally used to find new compounds, new bioactives. And that's what we did. It's part of it. We were a drug discovery group. So we were looking for new compounds from marine sponges. Okay. We go harvest them in the ocean, go scuba diving. It was a fun research, man. Bring it back to the lab. And, I did a field expedition to Papua New Guinea, scuba diving on like incredible reefs and walls and stuff that nobody had ever dived on before. And, so pretty exciting right at the beginning. So we were involved very much from the beginning on the harvest and then doing all the analysis back at the lab. So again, these combination techniques, hybrid techniques are useful, but it's not something you do as a routine. It really depends, again, what you need to get to in terms of a solution here. So let's talk about some emerging ones, which are and have been around, and none of these are like totally brand new. They've been around for a few years. DNA barcoding genetic testing.
So please talk about DNA barcoding. This has been very, very controversial in our industry.
It is. It's a highly advanced method, okay? And you can identify and authenticate botanical species because you're looking at the genetic material, right? And you're barcoding. So, but again, to do this, you need to have a reference library. Like you need to have a reference standard for HPLC. You need to have a reference library, a biological material that gives you the quote fingerprint or barcode of that particular plant species. So it can be very useful, but it does have its limitations. All right. So it's limited to known sequences. Now, I'm not a molecular biologist, and this is not my area of expertise, but and I've actually never done this, but you have a huge you're really susceptible to contamination. I mean, your sample prep is really key here. And again, it's very expensive. And again, it requires the molecular biology expertise. Okay. So, I think you referenced, I don't know when that paper was, it just got overturned. It wasn't
got overturned. Yeah, because one of the things that was problematic was this is an example of using a testing methodology that's inappropriate. I mean, like, let's say you have a reference standard for just ginkgo leaf, okay? Yeah. And you're gonna use that. You're gonna do some barcoding. Great. Now, let's say somebody does a ginkgo extract that's 50-to-1 extract, supercritical, and what you have left looks a whole hell of a lot different than the original ginkgo leaf. And if you're using a ginkgo leaf standard to do DNA barcoding for a 50-to-1, ginkgo extract that's highly standardized, it's not going to come out and go, oh yeah, that's ginkgo. It's going to give you, let's call it, for the sake of argument here, a false negative. It's going to say this isn't ginkgo, but it is. But you didn't have the right reference standard. You weren't doing it correctly, and it really isn't suitable for that. You should have been using that. You should have been using other kinds of quantitative methods for that. And that was the problem. And this guy did a bunch of these things, tested them completely wrong, and finally the journal, years later, overturned and removed it as incorrect. But it was Big thing at the time.
I
People
remember
said,
that.
"Oh, it's the herbal products industry. They're all a bunch of charlatans. They're selling you stuff that's not real." No, the testing method was not correct.
Well, and for the reasons I highlighted here, contamination or
Mm-hmm.
maybe not validated. And again, I think that was published in, what was it, 2005 originally?
Something like that. Yeah.
But the technology has advanced quite a bit now in the last 20 years for sure, and no question about that. But again, nothing is one off. There is no one size fits all solution. So, but it could be very useful in the scenario of checking adulteration, like if you're mixing another plant
Sure.
species in there. So like echinacea, you have echinacea purpurea and you have echinacea angustifolia, very different chemical profiles for both of those species,
Yeah.
right? And different research has been done on either one of them. And so, somebody could be calling it ashwagandha, by the genus species name, but yet they're cutting it or adulterating it with something else. Now, the DNA barcoding genetic testing could be a way to do that if you really want to do that. Well, let's be practical for a second, Gene. Most of these things, most of the ingredients that are supplied to product brands and the manufacturers in the industry have been doing that for years. So they control the supply chain.
Yeah.
If it's somebody, and again, most of the companies are reputable, we should say that. It's the one-off bad actor suppliers who you probably never heard of. I remember at the time of the elderberry thing, it came to my attention long before it came to the news because we were using elderberry in one of our products. And somebody was asking me about the supplier, and I said, first thing I asked, well, who's the supplier? Okay.
Mm-hmm.
And it's like the name of some company I've never heard of. And I said, are these guys distributors? Because you got people who are suppliers that are just brokers, distributors.
In fact, many of them are. There's some areas of the country where it's like distributor row, like a place in California where one building after the other, all they'll give you. We got 500 different things. It's because they get them from
Right.
None of them are from them. They get it from somebody else. They distribute them.
So again, you have to, when we talked about this before about when you're selecting a CDMO for manufacturing, it's the same thing with ingredients. You should be actively involved in your sourcing your raw materials and doing your due diligence on the ingredient suppliers to make sure that that's legit and they have very good qualifications that are robust programs that can make sure that they're basically selling what you said they're selling you, right? And they've done the testing and standardization there. But let's move on. We've got a couple more here to close it up on this. So FTIR, okay, what is that? That's Fourier Transform Infrared Spectroscopy. So this is a spectrographic technique. And I won't define what Fourier Transform is. It has to do with infrared light, okay, which has a certain wavelength. But basically, when you hit the sample with that particular wavelength, you're going to get vibrational energy. And this goes to physics and that sort of thing. You have different wavelengths, have different vibrational energies, and that's going to make the molecules in the molecular bonds vibrate at a different frequency and stuff. So again, this is a quick way to do it, but the biggest limitation to this is that, or I guess one of the major cons is that you have to have the right reference libraries where you've done a lot of testing. It's not a sensitive MS. It's again not a one size all fits solution. You can have overlapping spectra because again it's analyzing the extract or plant material in its full complexity. So if you think about a spectrograph, it's got like millions of peaks, but it basically gives you a fingerprint generally. Based on the different classes of compounds, you can get an idea of the fingerprint that looks generally like that. But again, it can be misleading if you think about the class of compounds of flavonoids, right? That has at least half a dozen different subclasses of those same category, the same skeletal structure that is ubiquitous in the plant kingdom. Okay, it's in many plants. So just because you identify a fingerprint in a certain plant species and you're doing another, you use the same method, it can look very similar. So again, having the expertise to do that interpretation and really knowing how to do that is really good. The last one we'll close up with this is metabolomics. Okay, and this is actually pretty cool. We were doing some of this with we were going to work with the university in Spain. This was just a couple of years ago, that was doing this type of research. And actually, we were going to look at saffron, right? Super expensive ingredient.
It's crazy.
It's crazy.
They've got the collection of that as a whole unto itself. It's so crazy. Yeah.
But we actually started this study, and where we were going to compare all the suppliers. Not that many suppliers, right? There's a couple key ones that have did the research or whatever, but there's some other lesser known that have a supply, but maybe they don't have all the research. So the metabolomics is a good way, really advanced analytical approach, that basically it studies all the metabolites or the small molecules that are produced by metabolic processes. Okay, so in botanical materials. So when you apply it to botanicals, you get a detailed biochemical profile, right? And it shows the diversity and the abundance of metabolites present in the plant. So really good way to identify the plant. So in this example of saffron, there's one supplier that's very expensive. I'm not going to call it any suppliers here. I'm just talking about the science. And they have their research on their extract. But when you compare the different extracts out there, they have different extraction methods. So that's going to change the chemical profile and theoretically would change its efficacy, right? Theoretically. Maybe it's not a huge change that wouldn't affect that. But using metabolomics would be a good way to identify. We never finished the project because the company didn't want to fund to do it, but it would have been a really cool way to identify maybe some lesser known extracts that delivered even better efficacy than the well-known ones, right? This is a positive way that it could be used to identify that. But again, it gives you, and it's in combination of chromatographic methods we talked about in the NMR. Okay, there's a combination of techniques you use together there, but you're looking at a lot of different components there in the plant. So again, it can give you a biochemical profile, like saffron has a certain biochemical profile of components. There's like 3 main components that are generally there. Mm-hmm. But again, it requires advanced instrumentation, expertise, and it's very expensive to do. And again, it's not something that you're going to do as a routine. This is like a research project, right? It's not like a routine botanical identity. You probably get there with TLC and HPLC, and you're good on most of these things.
So thank you for that. Now, what I've been kind of waiting for is I want to hear a little bit about the case studies. I think this is particularly interesting. So, come on, give us our boom, our zip, our zippity doo dah, our woo. There you go.
There we go. So I'll just share 2. There's many in my career, but I'll just share 2. And one goes back to 97 and one is 2023. So it really brackets, and there was a bunch in between. So it really brackets the whole approach of working with botanicals and also during that 25 plus year time frame, there's been a lot of advances in technology. We now have labs that have reference standards, can do to certain testing, whatever. You have USP has validated methods, but I'll just illustrate from beginning and more currently that we still have the same challenge. I don't care how advanced the technology is. You still need somebody with the expertise. The plant hasn't physically changed, right? It's still the same plant. It has been for thousands of years. So the chemistry profile is not changing. Maybe there's new methods for extraction to get a new extract or something. Sure. But the way we analyze these things, you still have the basic same approach that you need to apply here. And I think illustrating these 2 case studies is going to show that, and obviously over this time frame, you have people who've come and gone from the industry. You have new people in the industry who don't have the experience or previous knowledge of things like this, and so they don't know what they don't know. So we'll talk about some of my the learnings that I've had throughout my career. But the first story I'm going to talk about, it goes way back to 97. All right. And that is, I was director of R&D for a company called Pharmaprint. And nobody knows who Pharmaprint is, unless you were in the industry at that time in 97. It was a huge deal in the industry. And 1998 was the year that all of big pharma got into herbals or botanicals. Before that, Centrum, for example, right, or Bayer, They just did multivitamins. So vitamins and mineral products. That's it. No botanicals. They didn't touch them. They thought, oh, those are dirty. This is really what they said. Oh, those are dirty materials. We don't want them in our manufacturing facility. Okay. And this is, long before we had, the manufacturers we have today are very sophisticated. We've really evolved as an industry. But at the time, this was for Centrum Herbals. So there were seven herbal products, right? And it was 7 of the top 10 selling single herbal products at the time. So you had St. John's wort, echinacea, saw palmetto, ginseng, ginkgo, echinacea, and valerian. So, and those are still top selling botanicals
those are
today.
Great herbs.
Yeah, they all have their own research, and tons been done since then or whatever. But one of the unique things about pharma print, as PHARMA print, was that we did a pharmaceutical fingerprinting using NMR and Bioassay to determine raw materials, which ones were bioactive. Okay, so actually nobody's done anything like that in the industry. In fact, we put it on the label in the supplement facts below there that it had a certain bioactivity. So, for example, St. John's wort is known for, mild depression, right, or mood. And so we would screen the extracts. Of course, when this was announced, we were going to do Centrum. It was Whitehall Robbins at the time, or American Home Products. Everybody wanted to give us their St. John's wort, their echinacea, their garlic, their ginseng, whatever. And certain suppliers some of them who are still in the business today, supplied. I won't disclose that. If you ever see me at the trade show, whatever, I'll tell you who it was. But there's some interesting history here in terms of that. And one of these days, we need to write a book about the history in the business chain.
Yeah, that would be interesting.
It'll be we got some great stories, I'm sure. But let's stick to the science here. So, one of the ways we screened the extracts is we got samples of St. John's wort or whatever they're from several suppliers. And to illustrate the point we talked about earlier, we screened those with bioassay, and some had no activity. And it was like a panel of bioassays, right? It's in vitro bioassays that were pretty sophisticated. And so we were looking at maybe 20, 25 assays per indication, right? So, So for garlic was like cardiovascular health, St. John's wort, mild depression. So it'd be different screening, CNS panel, central nervous system panel for St. John's wort. Very fascinating work we did. And so we were able to screen the extracts and compare that with HPLC and NMR. And guess what? Not every St. John's wort extract was the same. Some had no biological activity and some had a lot. And and it tracked with the HPLC profiles, it looked very different. So some things were missing. So what was the reason for that? Well, we've already talked about some of the examples in terms of botanical sources. It can be seasonal variability when it was harvested. In fact, valerian fell out of the whole product launch because of supply issues, because it's seasonal to harvest, and they missed the window to harvest and they didn't have enough supply. This is a huge product launch. So it didn't launch. Although we did develop the product, we never actually commercially launched it. And by the way, everybody was racing to market in the consumer healthcare pharma space to launch their botanical products. So you had Centrum, you had Warner Lambert, who's not around. Pfizer acquired them. I believe it was Pfizer. And then, which was the Quantitative product. And they had all these celebrities, actors doing the commercials on TV. And then Bayer's one-a-day herbal. They had their one-a-day vitamin. Now they have their one-a-day herbal, right? These products don't exist on the market anymore, Gene, but it was...
No, I remember when they came out. It was a big deal.
It was a huge, huge deal. In fact, first year retail, we did $250 million in today's dollars. So I don't think I've seen a product launch like that for anything since then. So it was quite an experience. But anyway, so again, that illustrates one example in terms of really, we didn't investigate, what the cause was, right? But we wanted the best extracts that had the biological profile we wanted. But we didn't investigate what the cause was. It could have been adulteration. It could have been whatever, harvesting, seasonal variability, whatever. I mean, there is known to be a difference between the time you do harvesting of St. John's wort. You get a different chemical profile with hypericins and other components. So anyway, the other one's more recent, Jane, and that's, everybody knows KSM 66 ashwagandha. I mean, it's a major sponsor. Know the company really well. I've toured their farms and their manufacturing facilities. First class, vertically integrated, really control the quality. And they have probably the premier ashwagandha extract on the market. And they've been around. I think they launched that maybe 12, 13 years ago, at the trade shows and stuff, and they're a major sponsor. Everybody knows who they are. But the company I was working for at the time, and it was my EU colleagues, were looking at using this as a source in one of the other markets. It wasn't for the US, it was for Europe. And they actually, and this is like rookie mistakes, Gene, in 2023, that you'd think that we would have learned. And in fact, this was the impetus and some recent things that you and I know about of some other suppliers that we looked at, that these kind of rookie things continue to happen today. And again, it comes down primarily to people who don't have the experience or no knowledge of how to do this. The things I've been talking to, which is highly complex. It requires real expertise here. So,
and brings home your point about the need to validate the test method and use the right one.
Yeah, exactly. So in this scenario, just real quickly, a sample was sent. I wasn't directly involved, but I was overseeing and then advising, and they were getting a sample from Exariel, the KSM.
67 people. Yeah.
Yeah, they were getting a sample, and they tested it. They sent it off to approved lab, which they were using. And they came back that it said it had no withanolides, which is the main class of compounds in there. And I'm like, well, the C of A says it does. And I know for a fact they actually do the testing. And I've been to the lab in India. Okay, so I said, this makes no sense. So it's a real simple solution for me. I knew right away, they're not using the right test method. Mm-hmm. And investigated who the lab was. Well, they were using the USP method to test withanolides in it. And guess what? KSM66 is a proprietary extract, different chemical profile in terms of withanolides. And if you don't have the reference standards, which most labs don't, you can take the USP method and you're going to get the same result. It's not going to show any withanolides. But my colleagues didn't have the knowledge to understand that concept.
They didn't know what they didn't know.
Exactly. And I pointed out to them simply and said, okay, guys, let me prove it to you. So they have their approved lab in Europe and one in US that has the validated method, who has run it in their labs with the appropriate reference standards. And guess what? They tested the same lot of material and it got the result as expected that's on the C of A. So, it boggles my mind that people still today just think, oh, I can just go send this off to a lab and get it tested, and that's the result. They take it at face value. No, this is just a perfect example of inexperience, really don't know what they don't know, and don't have the privy of the history like you and I do, who have seen these things many, many times, right? I mean, now foods, right? They're pulling things off the market and testing for label claim. I cringe a little bit when people do this. It started with consumer labs back in the late 90s, after we kind of raised the bar on testing with Pharmaprint and Centrum and analytical methods. That's where Alchemist Labs started from, and all these other laboratories came out of that. And third-party testing, I helped NSF do their third-party testing program, right? So anyway, those are two good examples. You can go read the details of that in the article. It's fun to do. But we'll probably need to wrap this up and talk about kind of the key lessons learned here. What do you think,
some
Gene?
key takeaways, I think, would make sense.
All right. So here's the key takeaways. You can tell I'm passionate about this, Gene. I've been involved in this for many, many years. And I really wish people would, in labs and other folks would really listen to this and take it to heart, because they really need to talk to people like us that have the experience and stuff, especially if they're not getting the right results and stuff. So, and investigate it. Most of the times it's pretty doable to solve the problem and very easy to do that. So, so I think the first thing is you need to know, we said this already, you need to know the quality of the ingredients you're using in your products. Okay. You need to know the suppliers. You need to do your own due diligence, right? You can't accept everything at face value. I do know for a fact that some suppliers in the past, as a practice, would send you just a copy of a C of A, but not actually the testing they did on the lot that they shipped to you to use, right? Okay, that's a big no-no. Every lot should be tested, and you should have, and you should have the certificate of analysis from the ingredient supplier, and don't accept it at face value. Do your due diligence. You need a very robust supplier qualification program, okay? Otherwise, you're going to get yourself in trouble. Additionally, there's, we already said it, you need to have the proper validated test methods and reference standards, okay? So the KSM 66 story is a perfect example of that, where the laboratory did not have that and wasn't even aware that they didn't have the right validated method. And they thought, oh, I just run the USP method, it's good to go. This is just a complete ignorance in terms of natural products chemistry. Sorry. And it's by the way, it's a really well-known lab that everybody knows in the industry. So I'm not going to call them out on the carpet, but I was shocked. Okay, so, and you know, we have, and like I said, I cringe when I see these third-party companies. Now Foods is doing that. Hopefully they're doing the things that we talked about in terms of testing, and they're using the right validated method.
So far, it appears to me that they are doing just that. They appear to be using the right methods. They appear to be doing a good job,
right?
And I commend them on what they've been accomplishing there.
Yep, I do. And again, I think it's gotten a little lax in the industry. I think there's adulteration goes on a regular basis because it's supply and demand economics, and this puts pressure onto the supply chain, and it opens the door for opportunists who are going to try and take advantage of that. I mean, I can't tell you how many times I've been contacted by suppliers I've never heard of that has this, this, and this in their portfolio in terms of ingredients they supply, and I've never heard of them. Sure. So I get a lot of that. And
Me
so
too.
when I see those testing results, that third-party testing, by the way, there are third-party certification programs like NSF's program, USP, which is phasing out, by the way. The best one is really NSF. I'm a little biased there.
Yep.
Excuse me. But again, you know, anything we can do to raise the bar and call out the bad actors is good. But again, the first question I ask myself when I see these results is, okay, it's not why aren't they meeting label claim? It's what method are they using?
What method are they using? I've seen it. I've seen wrong methods being used and people being accused incorrectly of having a bad product, and just because of a bad test method.
Yeah. And so in our noble quest to raise the bar on quality and standards, to call out bad actors, let's make sure that we're managing that process well, and we're using appropriate test methodologies and labs that actually know what they know. Okay, so, right. And speaking of label claim, okay, you know, some of these examples that we've seen of products that didn't meet label claim, and by this different, goes right to the question of stability. Maybe they did test their product when they released it, but maybe they don't have a good stability program where they're testing it on shelf for 2 years of the expiration dating they're putting, right?
That's right.
So again, if as a product brand, you need to have a robust stability program. You need to be testing all your products to make sure so you don't end up a victim from some third-party testing initiative and end up in the news when you were, I venture to say, Gene, that some of these companies were shocked when it came out, and it's, oh crap, it was an immediate investigation internally into their, I know because I've seen this firsthand, into the quality, I won't say who, but into their quality standards and the methods they were using. So again, people get lax, they get comfortable, and this is something you have to stay on top of there.
You do. Well, look, so Bill, I just want to say, I had a number of questions, and you've certainly answered them all. And I just want to tell people again, read Bill's article. It's really, really good. It's pretty comprehensive overview, and and it's nice. And he's got some nice chart in there that you can look. What's the methodology you want to use for this? What does it mean? It's a really handy tool. So make sure to use that. So yeah, and then stay tuned. We'll be talking about from time to time other updates, developments in botanical testing technologies, and we'll do that. So to kind of wrap up, just thank you all for See, I'm in Tennessee now, so I say y'all. Didn't used to say y'all when I lived in New York. So thank y'all for tuning in,
Yep.
and make sure to like and subscribe. And Bill, you want to mention about your blog post?
Yeah. So again, I'll do a post on LinkedIn after our show once we have finished post production on the episode, and I'll include a link to the article. It's on my website. It's Nat Prologics. That's NATPROLOGIX.com. You can go to the blog there, or search my LinkedIn profile. You'll find it there as well. That'll link you back to it. So again, I'll share that for all our listeners, and really a good comprehensive review and updated with all the latest methods and the approaches. And a lot of it's just common sense, Jane. At the end of the day, it's just common sense. And if you don't have the expertise, find somebody who does have the expertise to help you in terms of if you're having struggles or identifying something. So that's all. My last message for that. And again, as Gene said, go check out our website, the bioactivenexus.com. So you can subscribe, follow the LinkedIn page, so on and so forth. And thanks for joining us today, Gene. Enjoyed the conversation. Something I'm passionate about, obviously, and I will continue to be passionate about because
I'm glad.
For me, at the end of the day, I know we talk about within our network of industry professionals is one thing, but for me, it's about the consumers because they're the ones that come up to me all the time and say, hey, Dr. Bell, what do I take for this? So I'm always concerned about the quality.
Always.
Not only is it meeting label claim, but is it actually going to work for them? Is there actually...
That's why when they come up to me and they ask, I will tell them what the nutraceutical is, and I'll generally recommend a brand that I'm familiar with, that I know that they do it right. Because otherwise, left to their own devices, they'll pick up some crap.
And that's a good inroad to do a partial reveal of something I'm working on a technology play in the industry that's going to be for consumers. That's really going to help them identify what are the right supplements for them and actually do deliver on the benefits that are claims. So I can't wait to reveal that when we finish that. We're in the prototype development right now. So that's all I'll say about that, but it's coming.
Very good.
Yeah, man. All right. Let's take it out. Everybody have a good week, and we'll talk to you soon.